Without further ado, I want to introduce our first speaker. So our first speaker, Abbey Myrick, is a PhD candidate in neuroscience at the University of New Mexico, where she studies how alcohol affects the brain, focusing on fetal alcohol spectrum disorders. A first-generation graduate student, she traded outdoor labor for a lab coat, a journey that has been as strange as it has been exciting. Curiosity brought her to neuroscience, partly to debug the brain, and partly because, well, drugs are fun, scientifically speaking. She is passionate about cutting through scare tactics to share real, useful information about how substances affect the brain. When not toiling away in her windowless lab, she likes to study how alcohol affects the brain, preferably on mountaintops, and can be found ranting about the patriarchy to her 2 dogs and 1 cat, or attempting to dance bachata with other humans. So, everyone, welcome Abbey Myrick.
Hey guys. My name is Abby. I study how alcohol affects the fetal brain at UNM, but I'm going to talk to you about how alcohol affects hopefully only the adult brain tonight. There we go. Okay, so the first thing that I want to touch on is that alcohol is a drug. This doesn't seem to be widely known, which is surprising, but alcohol is a drug, so we are all on drugs right now. If you are drinking alcohol, you are doing a drug. And I think this is really important to get our vocabulary correct. And I think to some degree the reason why we separate alcohol from all other drugs is probably due to the political history behind it. So before the Harrison Narcotics Act, which was the first major federal regulation of narcotics, drugs were kind of just up to the individual to decide when to take, how much to take. You could readily get narcotics over the counter for things like cough syrup or, as depicted here, for teething children. But in 1914, federal government started to regulate narcotics. And then in 1919, the 18th Amendment was ratified, and through the Volstead Act, Prohibition was enacted. Prohibition started in earnest in 1920, but this completely banned the sale of alcohol. And spoiler alert, it didn't work. So there are a number of reasons why it probably didn't work. One of them is that the states lost some income from the sale of alcohol. There was also an increase in law enforcement corruption as well as criminal activity around alcohol, because anytime you ban a commodity, you're going to have a black market pop up for it. And there was an increase in illegal speakeasies and the sale of dirty alcohol. So because it wasn't regulated, people were just making alcohol, bootlegging it, and the quality was very poor. Prohibition also came about as a response to World War I, and there was also a religious component to it to some degree, but a lot of it just stemmed from World War I and measures of austerity and trying to save money and save grain for the troops instead of making alcohol out of it. But so as time passed, these concerns kind of waned, and the 21st Amendment in 1933 repealed the 18th Amendment, and it's still the only amendment to repeal another amendment in U.S. history. Hopefully it stays that way. And so that ended Prohibition in 1933, and arguably around this time was kind of when the American drug war was born. We're all probably very familiar with the Nixon era, and certainly it snowballed in the 1970s, but this is when the federal regulations shifted away from alcohol towards narcotics and towards marijuana. So that's some of the history. So how does alcohol differ as far as how it affects our brain compared to other drugs? Alcohol is a very simple molecule compared to, yes, we went through this. Compared to all these other substances that are generally put in that category of drug away from alcohol, molecularly, alcohol is very simple, and this is important in regards to how it affects the brain compared to these other drugs. So we probably know more about how opiates affect the brain than we do about how alcohol affects the brain. And the reason for that is that alcohol's effects are extremely widespread throughout the brain. and that is related to how simple its molecular structure is. So on the cells in your brain, you have these receptors, and if you have, it's kind of like a lock and key system. If you have a very simple molecule, it can fit into the lock and key of your receptors in the cells, and it can affect the receptors, which affects the cells. But if you have a very complicated molecular structure, it's harder for that molecule to interact with the different receptors on your cells. And so you have actually end up with a narrower path of like influence from an opiate compared to alcohol. So we refer to alcohol as a dirty drug because it affects basically the entire brain. So talking about neurons, neurons are the cells in your brain. And there are other cells in your brain, but I'm only going to talk about neurons. So hopefully, if there's any astrocyte lovers out there, don't come at me. I'm only going to talk about neurons. So the way that we classify neurons, we kind of use two different measures. One of them is what kind of neurotransmitters do they use overall? So do they end up being excitatory or inhibitory? And you can think of neurotransmitters as little like packages, and one cell will shoot a package across the void to the next cell, and the next cell will receive that package via its receptors. And if that package is excitatory, then that will cause that receiving cell to fire itself and continue on the signal. If that package is inhibitory, then it quiets that receiving cell down and that signal stops. And we also use morphology, so the shape of the cell, and these two things combined are kind of generally how we classify neurons. And there's a whole bunch of different kinds of neurons in your brain, and they all have this amazing like branching tree root shape to them. There's a lot of different, morphology within that, but they're all just like really beautiful. So the receptors on the cells, you have endogenous neurotransmitters. So those neurotransmitters develop to interact with their particular receptor. And so it's like a circular peg fitting into a circular hole. But then if you have like a random drug that it's not developed for that receptor, it's just like not gonna work. But alcohol is like small enough and simple enough that it can seemingly find somewhere on that receptor to interact always, and so it has some consequence for the cell. I don't remember what paper I got this diagram from, but I really liked this guy, and I'm sorry I forgot to cite it, so if you're the author of this paper, I apologize, but I took this diagram from an ataxia paper. So at the beginning of Nerd Night, you're feeling fine, maybe you have a couple of beers, and then at the end of Nerd Night, you're kind of like a little bit wobbly maybe. And hopefully if you're feeling this way, you have a sober driver. But what's happening here? Why is alcohol affecting movement? Well, I'll preface this with the fact that I'm going to talk about two particular regions in the brain, but the brain is like the most complicated system that we've ever discovered as humans. And we're all just like walking around with one, which is insane. But there's no region in the brain that works in isolation. So there's always like feedback from other regions, so we can isolate a particular zone and know that this is really important for one thing, but it's also important to remember that it's always working in concert with other regions in the brain. So the cerebellum is very important for fine motor coordination, and I wanted to talk about it because it just looks really cool. It's called the cerebellum, which means little brain, because it's basically just like this extra little brain behind the rest of your brain. And it's really neat looking. I had the opportunity to dissect a human brain, which was really amazing. If there's any donors in the audience, thank you so much. It's very helpful. So on the outside of your cerebellum, it just kind of looks like a bunch of folds. But then when you cut into it, you see that each one of those folds looks like a fern frond on the inside, or like some kind of underwater plant. They're just absolutely beautiful. And presumably, all those extra folds allow for increased surface area, which means more cells can be packed into the same space. That's also why the rest of the brain has that sulci and gyri, the wrinkles on the cortex. So just tons of cells packed in there. And the actual, one type of cell that's in the cerebellum is also really cool looking. It's called a Purkinje cell. And I'm just going to nerd out a little bit on where this diagram came from. It was drawn by Santiago Ramón y Cajal, and he was a Spanish artist turned medical researcher, and he was born in 1852, and he took one of the first staining method that we had to visualize neurons, and he ran with it and developed it further and used his artistic skills to look through the microscope and draw out what he saw, which were just absolutely beautiful. They were the first images that we had of cells in the brain, and his drawings are really impeccable. I have some of his retinal neurons tattooed on my arm. I also got to go to the museum in Madrid and see some of his original works when I went there for a conference last year, which was really incredible. But yeah, his drawings are just amazing. Anyway, so Purkinje cells are inhibitory. And just in case you don't believe me, those are real cells that are stained and visualized under the microscope. They really do look like that. They're absolutely crazy. Okay, so how does the cerebellum, what does it have to do with movement? So you've got the cerebellum, you've got these Purkinje cells, and obviously this system is much more complicated, but I'm going to focus on one aspect of it. So I tried to think of a good analogy, and I think in New Mexico, maybe like salsa dancing and flamenco might be a good one. So, you know, like if you've ever done that kind of dancing, you have to keep like a frame. You and your partner have like this push and pull where you're constantly applying either a push or a pull to maintain that distance and maintain that frame. So when there's a sensory input to the cerebellum, it's believed that the Purkinje cells sort of act in a way that they can mediate their output. They're like the main output for the cerebellum based on the strength of that sensory input. And what that means is that they sort of act in this push and pull manner so that the output, your movement in response to some sort of sensory input, is mediated. And so you get a smooth movement instead of like jerky or spastic. Purkinje cells are affected in Parkinson's disease, and the cerebellum is actually really sensitive to alcohol. So if you have an alcohol use disorder that's quite bad, your Purkinje cells can get damaged and you'll develop ataxia, which is that jerky movement. Okay, so what happens with alcohol on board in the cerebellum? Okay, so we got cerebellum, we got some alcohol on board. You normally have this system where it's like two flamenco dancers that are doing this beautiful dance, creating this smooth movement. But we know that alcohol actually inhibits the function of Purkinje cells and the cerebellum. And so this sort of like releases the brake, and you get these jerky, discoordinated movements. So your fine motor movement goes away. And so you might end up, you know, spilling some beer, and again, hopefully if this is you, you have a sober driver. And I just had to put a picture of Lucille Bluth, because she's always what I think about when we talk about the effects of alcohol on motor movement. I heard some laughs, so there's some millennials at least in this audience, but if you haven't seen Arrested Development, it's a fantastic show. Okay, and so these effects exist on a spectrum, right? Impaired coordination all the way up to like you're falling down and getting a concussion. Clearly dose affects this. If we drink more, we're gonna get more affected. Tolerance also affects this. So if you have more practice drinking alcohol, you might not see these effects as readily as somebody that's alcohol naive. And also rate of consumption affects this. So if you do a keg stand, you're going to be a lot more messed up than if you drank that same amount of alcohol over like a couple of hours. And the reason for that is directly related to overloading the enzymes that are mostly in your liver that break down alcohol. And interesting fact, that varies from individual to individual, how many of those enzymes. Oops. Somewhere. There we go. Okay. Okay, so that's movement. So now I'm going to talk a little bit about memory. And this is a little bit, so we know that alcohol affects this thing called long-term potentiation, which is a component of learning and memory. But I study sharpwave ripples, so I'm going to talk about that because I think it's the coolest, and this is Nerd Night. We're supposed to nerd out about the things we're nerdy about. So I'm just going to preface that. Okay. Clearly, we all know that alcohol affects memory, right? It saturates our like pop culture. We have so many movies about this, like I drank too much, I don't remember what happened, hijinks ensues. But what's actually going on? So one of the regions that's really important for learning a memory is called the hippocampus. It's essential for forming new memories. We know that. It is absolutely essential to the formation of new memories and the consolidation of those short-term memories into long-term memories. It's called the hippocampus, which means seahorse in Greek, because it looks kind of like a seahorse. And so there's pyramidal cells, which are excitatory in the hippocampus, and there's also interneurons, which are inhibitory. And just my man Cajal, again, bringing back the poetry into neuroscience, he once referred to interneurons as the butterflies of the soul, which I just love that. They do kind of look like butterflies. He thought that they were particularly important for human cognition and the development of our human brains because there's an abnormal ratio of them in the human brain compared to other mammals. Anyway, okay, so you've got information from the outside world coming into the hippocampus. The hippocampus has these really cool oscillations called sharp wave ripples. And I drew a little diagram up top there, example, where you can see the sharp wave followed by that high frequency ripple. And when we talk about an oscillation, we're talking about the summation of many, many cells in that region firing at once, and they create this voltage trace. So we're looking at a voltage trace. And then underneath that is an example of a roster plot where each row is a neuron and each hash mark is where that neuron is firing in that oscillation. And so cells will have different patterns of firing that vary between sharp wave ripples. And we think it's kind of like Morse code, where like that different pattern encodes different information for each memory. This is an example trace from a mouse that I recorded from, so you can see two sharp wave ripples there in blue. Okay, so the pyramidal cells are kind of like the gymnastic ribbon dancer. They're like flying in the air, and then the interneurons are like, no, you gotta chill out. And it's sort of this interplay of those two types of neurons that create this ripple that you see. And importantly, there's a lot of different kinds of learning and memory. I find this absolutely fascinating. So there's this really well-known individual called HM. You guys have probably heard of him. He's in like every neuroscience textbook ever. But he had part of his hippocampus removed, and he couldn't make any new memories, but he could remember everything before the surgery. So all his long-term storage was still there because it's a different part of the brain that's responsible for those long-term memories being stored. He also was able to actually develop new motor skills. Even though he could never remember practicing that particular motor skill, his ability would become better and better every day that he did it, even though he never remembered doing it before. So this tells us that different regions in the brain are responsible for different types of learning and memory, which I think is really fascinating. Okay, so just to recap, you've got these sharp wave ripples, excitatory pyramidal cells, inhibitory interneurons. They're firing in particular patterns to encode different types of memory or different memories. And then they get replayed over and over, particularly during sleep. And the hypothesis is that the repetitive pattern of sharp wave ripple is somehow strengthening that memory, but also moving it out to the cortex for long-term storage. And we call that consolidation. Okay, but then what happens with alcohol? So you've got information coming into the hippocampus, but now you have alcohol on board. Normally you've got this nice ripple oscillation that's carrying information about what I'm saying. But then when alcohol is on board, we know that it inhibits the pyramidal cells. So it keeps them from being able to fire while it potentiates the interneurons, so they're firing more. And so this is skewing the ability of these cells to participate the way that they normally would. So in theory, that would be disrupting the Morse code pattern that you would normally see. And so you might forget by the end of nerd night, depending on how many drinks you've had, where you put your beer or, whatever. And so again, this exists on a spectrum all the way from impaired memory formation all the way up to blackouts. And blackouts are really quite crazy. But again, this is affected by dose. It's affected by tolerance, although it's a little bit more gray area as far as how compared to motor movement and the rate of consumption. So I just kind of want to like revisit this idea of a blackout because I think it's really interesting. So you might have enough alcohol on board that your cerebellum is affected. So like, you know, your movement is not great, and like another part of your brain is affected enough that you're slurring your speech, but you're still able to talk and you're still able to walk. And so your friends might be like, hey, do you want to go to the bar down the street? And you're like, yeah. And then like you get there 5 minutes later, but then you don't remember like ever saying yes to going to the bar, and you're like, why are we here? Well, that would indicate that your hippocampus has enough alcohol on board that you can't form those new memories. And so this is how we're able to see where the body is functioning, but there's no recollection of what happened. And arguably, even in the moment, maybe within minutes, seconds, we don't really know because this is really hard to study, a person might be able to say yes to something, but then not really like remember 5 minutes ago when it happened. And so this is how we get this sort of weird phenomenon where somebody is able to function bodily but can't remember. So if you ever see somebody in that situation, help them out. All right, I'm happy to take any questions. I'll try to be more helpful than Lucille Bluth, but thank you so much for listening.
Are there any other sorts of drugs that can interface with the brain as closely as alcohol can, or is alcohol pretty much the king of interfacing? Maybe repeat the question.
You mean like affect so widely, or?
Yeah, like I guess when you're talking about like a key fitting into a particular slot, are there any other types of drugs that have such a broad application of where they?
Yeah, the question was, are there any other drugs that have such a widespread effect on the brain? To my knowledge, no. To my knowledge, but I only study alcohol. Yeah.
This is more of like a cultural question, which I don't know if you can answer, but
Yeah.
at what point did we figure out that alcohol was bad? And like, it's been so, like, we can trace back alcohol consumption to like Mesopotamia.
Yeah.
It was a method of like preservation, right? Every fermentation byproduct of alcohol is present.
Yeah.
Like, is it because it's so culturally ingrained that we'll never eliminate?
Yeah, it's a great question. So she was asking, do we know like when people started to realize that alcohol was bad for you? And yeah, I can't like definitively answer that, but I think there's probably multiple components where like people in like long ago, they didn't live as long, so we started living a lot longer. So that's something that comes into play with a lot of like medical science, where we're like, oh, we're actually living to be like in our 80s, 90s, 100s, and some of these things are affecting us badly that we didn't realize before. But yeah, I can't answer like specifically when people would have started realizing like the effects on the liver and stuff. I want to say probably at least the 60s, but you would think it'd be even earlier. But yeah, sorry, I can't give a definitive answer. Yeah. Yeah, that's a good question. Is there any evidence that like training your memory can help you resist alcohol, like the loss of memory due to alcohol? I don't. I haven't heard of it, and like from a scientific perspective, I would find it hard to imagine. But maybe, I mean, if your memory is already stronger, then like maybe the deleterious effects of alcohol would be like, you know, a little bit, you know, less overcoming, I guess. I don't know. Again, it's very dose dependent too, right? Like if you just moderately consume alcohol for your whole life, probably strength training your memory like is gonna help a lot. But if you're like talking about consuming large amounts, then you're probably not gonna see an effect there, I wouldn't think. Anybody else? Yeah. No, cannabis is a completely different, different receptors, different drug, different action. I don't research cannabis, but yeah, it's definitely a new field that people are trying to figure out what's going on there too. Sorry, can you repeat that?
What type of scans do you do?
Scans. So I work in preclinical research, so I use mice, which I'm able to implant electrodes and actually measure the voltage change in the brain. We can't do that with humans for the most part. There are actually humans that have probes in their brains for other reasons, and they contribute a lot to our understanding of how a lot of this stuff works. Scans, like if you're thinking of like an MRI or something, those are helpful to, those have shown us like how much mass can actually be lost in the brain due to like an alcohol use disorder. But for the most part, a lot of like our mechanistic understanding of how alcohol works is coming from preclinical research. So that's like animal research.
People or mice can what? Oh.
I don't know if I've ever heard of somebody being born without their cerebellum. I mean, maybe I don't know how functionable that would be after the fact. Sorry, yeah, I don't know. I'm sorry. Is there another? Sorry.
Is state-dependent learning with alcohol a real thing?
No. I've never seen any scientific evidence for it. And it just doesn't really, sorry. He asked if state-dependent memory is a thing with alcohol. So the concept of like, if you experience something when you're on alcohol, if you get drunk again, would you be able to remember it just because you're back in that same state? As far as I know, there's no scientific evidence for it.
But I remember the effects of tequila. The tequila that I drank.
Uh-huh.
About that.
Yeah, preach, girl. I don't know. Yeah.
Out of since alcohol interfaces with so many different parts of the brain, and THC, for example, is one that seems more complex. Like, are there any molecules you've seen that are naturally occurring that are particularly safer to interact with, and lower rates of long-term effects?
Are you asking me what you should get high on other than alcohol? Okay. I cannot answer that question.
Like, I guess I'm kind of wondering if there's like, for long-term effects-wise, have you noticed any other types of drugs that people probably intake into their system that don't have as much long-term? Alcohol is obviously.
Yeah, so I get in trouble for saying this sometimes, but from a neuroscience perspective, opiates actually are way less damaging than alcohol appears to be. But you have to weigh that with it's also vastly more addicting for some people. Again, individual physiology plays a huge role in it. Genetics plays a huge role in it. It's upsetting. I deal with mice, right? And we have strains of mice that love alcohol. Like, they love it. They will drink so much of it. And then we have strains of mice that don't want anything to do with it. They hate it. So it's very obvious that genetics plays a big role. But yeah, I can't really advocate because it's such an individual thing. Like, one person can smoke marijuana and be fine and like not really have an issue, and then another person, like, it can really interfere with their life, even though it's a pretty chill drug for most people. Same with alcohol, really. And then there are actually cases of people shooting heroin for a couple years and then stopping. It's not always a cut and dry thing, and unfortunately in research we tend to focus on researching the worst case scenario instead of also researching what happens when somebody can use something and not develop a problem. So there's more people are getting behind researching that to kind of see like what's the difference in people's brains and physiology that allows them to walk away from a substance instead of developing a substance use problem. Yeah. Are there any definitive vitamins or minerals that can help you learn? I'm unfortunately not qualified to answer that. I don't know. It's a really good question. I know that thiamine can help with some of the negative effects of alcohol when you have like a lot of alcohol, but that's not really like specific to learning and memory. So I'm sorry, I can't really answer that.
Like following up on my cultural question, have you seen in your research like ethnic subgroups where the addictive qualities or maybe the enzymatic reaction to alcohol is due? I mean, you can say genetics, but you can look at their cultural history and the consumption of alcohol.
Yeah.
Historically, is there like a correlation, or is it kind of, if you actually look at it more holistically, it's actually pretty randomized based on your really unique genetic makeup? Like I think of like South Asian subcontinents versus like Nordic countries, right?
Yeah. Are you asking not just the effects that alcohol has on the system, but like how culture plays into?
Not really culture, but like could you,
Okay.
I guess culture is in a sense like Southeast Asians, although we have alcohol, the history of consumption
Yeah.
is not as relevant as like the consumption of low to mid alcohol beers and
Yeah. Yeah.
short, like we're talking historic.
Yeah. The
So then things like liver disease or brain abnormalities might be more prevalent or more susceptible because of the genetics. I don't know if that's that. Is that true or is that
The only thing I can say in regard to that is that if you've heard of like Asian flush, where people of Asian descent who drink alcohol will get very flushed and often don't feel good after drinking alcohol, that's related to low enzyme count for what breaks down alcohol. So that plays into those general populations not consuming a lot of alcohol. But as far as like, I don't think there would ever be a pool for that where people would drink a lot of alcohol because it makes you feel bad. So I don't know if I'm understanding your
like
question
having a
correctly.
lineage, let's say I have a lineage
Yeah.
of, like consuming alcohol.
Yeah.
And then I come out and I'm genetically modified to, well, because then I'm, are you building resilience?
Oh, are you like
Generate?
building immunity?
I don't know if immunity is the right word.
Yeah. Yeah, I don't know. That's a good question. I don't think that doing the drug would affect well, actually it might. It might. You like,
And I'm not talking about like my parents, I'm talking about like generations.
yeah, like if you kind of like had generation after generation that was drinking a lot, would you sort of like develop more enzymes basically and be able to break that down? That's a good point. Yeah, it's highly likely, but I don't know any research around it. No.
Thanks, sorry, that was
No, we got there. Sorry, I'm bad at comprehension. Oh. Yeah, that's like your prefrontal cortex. Yeah. Yeah. Yes. All right.
Awesome.